So good morning everyone. This lecture we will discuss respiratory drug development, mainly beta agonist. Okay, not all the different types of respiratory drug development, just the beta agonist. So at the end of this lecture, you will be able to explain where and how respiratory drug works and describe the principle of how respiratory drugs are designed, mainly when you say respiratory drugs, mainly beta agonist, okay, beta 2 agonist. And then you will be able to explain the process of how respiratory drugs are designed, illustrate the respiratory drug design and development using example, and you'll be also able to explain mechanism of action, SAR, and when you discuss the SAR, you will know the uses and reasons for side effect of various respiratory drugs. So this chapter you will find very interesting. Okay. So before we go to the chapter, let us recall certain common facts that you might have studied already in pharmacology. So it's important to have recall to get connected. So I hope all of you know what is adrenergic receptors, right? They are G protein coupled receptor, which consists of 7 transmembrane helix, right? And there are two types of adrenergic receptor, that is alpha and beta, and 3 of these helix are involved in the binding site of beta adrenoreceptors. So do you understand? Do you have clear idea about the transmembrane helix, 7 transmembrane helix, what it means, and the loops? I'm wondering to know.
No.
None of you?
For me, no. No.
Okay, let me do one thing. Let me go to the slideshow, because in the slideshow I can use my pen. I will draw something and then I will show you all. Let's check once again today whether you can see the slideshow or not. So I have created a blank slide. Are you able to see it?
Yes.
But it's not slideshow.
Okay, I will make it, don't worry. Okay. I'm drawing something, then after drawing I will keep the drawing. Nobody see, I'm drawing the 2 cell membrane. Nobody can see my drawing?
No.
No.
Okay, don't worry. 1, 3, 6, 7. Okay. C double O H. One, two, I'm drawing something. Please have patience, okay? I will be back to you after I finish my drawing. I will show you the drawing. So this is known as TM1. I hope you can see this in your videos. TM1, 2, 3, 4, 5, 6, 7. Good, I'm done. This is intracellular, extracellular. Intracellular. Okay. Done. So nobody can see once again. No one.
No.
No.
Nothing. Main clear.
Okay.
Yes,
Okay,
now
now
we
it's
see
down.
it.
Yes, we saw it.
That's good. Okay. So see, what did I say here? G protein, they are coupled to the G protein, okay, which could be inhibitory protein or stimulatory protein. GTP binding protein. So we will not talk about that. Just need to remember which consists of 7 transmembrane helix. 7 transmembrane helix. What are those 7 transmembrane helix? Look at this slide. You see, these are all transmembrane. You see the yellow color line. These are the cell membrane. Okay, the 2 cell membrane. So transmembrane means it is neither completely inside the cell nor completely outside the cell. You see, I have written on the right hand side extracellular and intracellular. Below portion is the intracellular region. Above portion is the extracellular region. Okay, so these receptors are transmembrane receptor. That means they remain in between the membrane, neither completely inside nor completely outside. Some part will remain outside, some part will remain in between, some will remain inside. So see, every receptor, they are made up of amino acid. So that means they will have C terminal and N terminal. So the N terminal, that is the amino group, N terminal will remain at the extracellular region. You see the N terminal here. Can you see? Once again. Can you see the N terminal, all of you? Yes or no? Keep
Yes.
responding. Amino end, you see. So from here it starts. So it goes down, then comes up again, then again goes down, comes up and goes down like this. So this way, how many helix? You see the transmembrane helix TM region 1, 2, 3, 4, 5, 6, 7. Total 7 helix, right? So it has 7 transmembrane helix. These are the helix. So if you see, it has how many loops? Can you tell me? See the helix 1, transmembrane helix 1 and 2 is connected by intracellular loop 1, that is IL1. Can you see at the bottom IL1?
Yes.
See, this
Yes,
is IL1.
3 loop.
Yes.
Yeah. So there are 3. That means you have 3 intracellular loop and 3 extracellular loop. You see the transmembrane helix 2 and 3, they are connected by extracellular loop 1. So in the structure, total how many extracellular loop and how many intracellular loop you have? Hello?
3 intra and 3 extra.
3 intracellular loop and 3
3
extracellular loop, correct? Which is connecting the 7 transmembrane helix. So you have clear idea about how the receptor is present inside the cell, right? Is it clear, all of you? And the C terminal region is present inside the cell. You can see C double bond O as COOH. That is a C terminal carboxylic end present inside the cell. N terminal present outside the cell. Okay. So is it clear? Can I go to my next slide?
Yes. Thank you.
This is the common structure of adrenergic receptor. So they are G protein coupled receptor, consist of 7 transmembrane helix, and there are 2 types of adrenergic receptor, that is alpha and beta. And among the 7 helix, it is the 3 of the helix, that is TM3, 5 and 6, which are involved in the binding site of the molecule drug. So remember, this is the key information again. Which helix are involved in binding to the drug or the agonist?
3, 5, and 6.
3, 5 and 6. Now, one more thing you remember, the helix that you see 1 to 7, I have shown it in 2D structure. So they don't remain like this, separate from each other. In the 3D, they will undergo folding. In the 3D structure, tertiary structure, they will undergo folding. This TM3, 5 and 6, they remain close to each other. Okay, they remain close to each other. And so the binding site will remain, they will form a binding site like a pocket. Binding site means pocket of the receptor where the drug or the internal agonist molecule will bind. Okay, I will try to show you a 3D structure in the software, maybe in the next class. Just remind me. So see, adrenergic receptor alpha and beta can be further subclassified into alpha 1, alpha 2. Beta can be classified into beta 1, beta 2, and beta 3. Beta 3 is still less explored. There are many hypotheses though, but mainly beta 1 and beta 2. Similarly, alpha 1 and alpha 2 also can be subclassified into different classes. You need not to know all this thing. Just remember beta 1, beta 2, alpha 1, alpha 2. Now regarding the signal transduction, when they are stimulated, okay, alpha 1 adrenergic receptor, they generate inositol triphosphate and diacylglycerol, okay. And whereas alpha 2 receptor, they inhibit the cyclic generation of cyclic AMP, and beta receptor, they generate cyclic AMP. Cyclic AMP, you know, they are the secondary messengers which perform various functions. Now we go to the next slide. In this slide, try to understand this. You might have studied again. In this slide, we are trying to understand the location of this receptor and how they influence in show their different effects in different part of the body. So first of all, you see in the first column, that is the organ or tissue. So in the heart muscle, it mainly contains the beta 1 receptor. So when you stimulate beta 1 receptor, it results into muscular contraction, okay, which in turn causes increase in heart rate and force of contraction. So this is very important. You need to remember. Now, secondly, you see, when we talk about the bronchial smooth muscle contain both alpha 1 and beta 2 receptor. So when you stimulate alpha 1, it causes smooth muscle contraction, whereas and beta 2 also. When you stimulate beta 2, it causes smooth muscle relaxation. So contraction of smooth muscle will result in closing of airways, and relaxation of the smooth muscle will cause the dilating and opening of the airways. So what you need to remember and observe from this here, you see two receptor, beta 1, okay, which is present in heart, beta 2, which is present in the airways, okay. The stimulation of beta 2 causes relaxation, whereas stimulation of beta 1 causes smooth muscle contraction. It will increase the heart rate and force. Now, listen here. How do you remember why we need to study this? Because when you give a drug, since this receptor has similar topology, okay, they are structurally they have a lot of structural similarity. So there are chances. The many drugs which are non-selective, they can act both on beta 1 and beta 2. So under such condition, imagine if a person is suffering from asthma, okay, and if you give a drug, and if the same person is suffering from heart disease, and if you give a non-selective drug, what will happen? It may stimulate the beta 2 receptor. Yeah, not bronchospasm. Okay, I repeat my question. Imagine a patient who is suffering from two different conditions, having the asthma and also having hypertension. What will happen if you give a drug that is non-selective to beta 1 and beta 2?
It will cause a heart failure.
It will
cause a heart failure.
Yeah, so it will. So when you give the agonist, which is non-selective, it will not only help in relaxing the smooth muscle of the airways and dilating the airways, but it will also increase the heart rate, because stimulation of beta 2 causes the muscle contraction, increases the heart rate, right? So when you give a drug, it must have to be selectively binding to beta 2. Now at the same time, if you give a drug and if it non-selectively bind to alpha 1 and beta 2, what effect you will see? If the drug non-selectively bind both to, I will enlarge the slide. Okay, if the drug non-selectively bind to both alpha 1 and beta 2, what effect you will see? Alpha 1 and beta 2 of the bronchial smooth muscle.
It will increase the pressure, the heart, blood pressure, cause hypertension.
You did not get my question. My question is alpha 1 and beta 2. I did not ask you about the beta 1. My question is, if you give a drug, if it is non-selectively binding to alpha 1 and beta 2, of the bronchial smooth muscle, what effect you will see?
They are opposite to each other, so no
Nothing.
effect.
Nothing.
No
Yeah,
effect.
so since they are opposite to each other, you will not see any proper effect. So that means a drug has to be selective. If we want to dilate the airways, we must give a drug that must be selectively binding to the binding site of beta 2 receptor only, neither to beta 1 or not to alpha 1. Yeah.
Doctor, is there any drugs that act on alpha 1 and beta 2 at the same time?
We will discuss, we'll come to the drug later. Okay, now try to understand what happened. Yes, there are. First of all, to answer your question, yes, there are drugs. When we since see the name of the chapter, respiratory drug development, that means we'll see.
No, I mean the same drug.
Yes, there are drugs which
Act on both.
which can act on both. We will see it later. Okay, later part of the slides. So what we are trying to understand the importance of selectivity. Now, similarly, if you see the distribution, the beta 2 is present not only in bronchial smooth muscle, it is present in arterial smooth muscle, which causes smooth muscle relaxation. It present in veins, it causes smooth muscle relaxation. So activation of beta 2, okay, or the agonist of beta 2, it will always result in relaxation, whereas the activation or agonist of beta 2, beta 1, it will cause contraction. All right. Now, similarly, they are present in other part of the body also. For example, beta 2 is also present in kidney, which activation of beta 2 will result in increase in renin secretion, which in turn again may cause increase in blood pressure. All right. Now, based on this, I want to ask you a question. Let me see, think properly, and let me see who can answer it properly. See, so now beta 1 is also present unfortunately in kidney, and activation may cause renin secretion, and you know the renin can increase the blood pressure by converting angiotensin 1 to angiotensin 2. Those things we'll discuss again in different chapter. Now my question here is the basic concept. Listen carefully. What do you understand if I ask you what is the meaning of selectivity of a drug? The question sounds simple, but let me see how many have clear idea.
Selectivity on which receptor to works on which organ.
Yeah, can you again explain this one? Selectivity means what?
To work specifically on a type of receptor in the organ.
In the organ, what do you mean by organ? In the organ?
As in the liver or the heart or the muscle.
Yes. So, yeah.
Target specific.
Yeah. Would you like to?
Without another organ.
You need to work on organ and not on others?
Yes. Specificity means not only the selective, okay, to a particular receptor. So here, how many receptor we are seeing? We are seeing three different types of receptor: beta 1, beta 2, alpha 1, right? So selectivity means selective to particular receptor. This is the first point. And also not only to particular receptor, but particular receptor of a particular organ. Okay, that is the complete selectivity. You understand or not? So selectivity does not only mean to a particular receptor, but also it means the receptor of a particular organ. Okay, so site specificity and receptor specificity, 2 points comes to specific. Otherwise, you see beta 2 is also present on kidney, right? So if it acts on kidney rather than what is that, airways, or along with airways, it will again result in increase in the blood pressure. Yes or not?
Yes.
Yeah, so the selectivity means organ specificity and receptor specificity. It can be enzyme. When you talk about enzyme, with the condition applies the same. Now, it is always a challenge to discover and develop a drug which is specific to a particular receptor, and that too specific to particular organ. That is why most of the drugs, they show their side effects because of off-target activation of receptor or a particular enzyme, off-target tissue. That means rather than activating or acting on a receptor of a particular tissue, it will also act on the receptor of other tissue where it is not supposed to act, because of which you see a lot of side effects. Okay. So with this understanding, I want to ask you one thing. Often I have seen that many students, they get confused. At the end, they don't remember which receptor is present in which organ. So from here, even though I have given a list of distribution of different other receptor, we will try to remember only beta 1 and beta 2 for this chapter. Now, how do you remember which receptor is present in heart and which one is present in the lungs, airways? Any clue?
Beta 1 in heart, beta 2 in the lungs.
Yeah, any simple way to remember?
When you beta 2 is like two B letters. They look like lung.
No. See, simple way. How many lungs?
Two
lungs
lungs and
beta 2. Yeah. How many heart? One heart.
One.
Right? So beta 1. Very simple way to remember. Okay. Although there is no scientific logic behind, this is a kid's way of remembering. One heart, beta 1. Two lungs, beta 2. Okay. Don't get confused, because next chapter we will be discussing about the beta blockers. Okay. So this chapter we will be discussing the development of beta 2 agonist, because we need agonist to activate selective beta 2 agonist, okay, to cause the relaxation of smooth muscle of the bronchial airways, okay, so that it can dilate and open the airways and help the asthmatic patient to breathe. We go to the next slide. So see the distribution and effect of adrenoreceptor in different part of the body. Activating alpha adrenoreceptor, what happens? It causes generally contracts the smooth muscle except gut. And activating beta 1 receptor contracts the cardiac muscle, whereas activating beta 2 adrenoreceptor relaxes the smooth muscle, opposite to each other. And beta 1 adrenoreceptor is predominant in heart, whereas beta 2 adrenoreceptor is predominant in airways. And as I have discussed just now, these receptors are distributed differently in different organs and tissue. Okay, and receptor selective drugs act selectively at different organs and tissue. So here we discuss 2 points. When we say receptor selective drugs or selective drugs, means it has to be specific to a particular receptor and plus particular tissue. Now going to the next slide. There are two different chemical messenger that acts on this receptor. First one is a noradrenaline, which is also known as norepinephrine. It's a neurotransmitter released by sympathetic nerve, which feeds smooth muscle and cardiac muscle as well. Okay, look at the structure on the right hand side. Then adrenaline, which is a hormone released from the adrenal medulla and circulates in the blood supply in order to reach the adrenergic receptor. If you look at this, both the structure, they have something, epinephrine and norepinephrine, or adrenaline, noradrenaline, they have something in common. What is common in them? They both have a catechol ring system. Okay, so they and a alkyl amine side chain. That's why they are categorized under a class of compound known as catecholamine. So let me show you. You know what is catechol group? What is catechol group? Who can tell? I want to see how many of you have the clear knowledge on this.
OH.
Benzene ring with OH.
Is it the ring with OH only?
Benzene ring.
Okay. Benzene ring with 2 OH, you mean? If I say no?
Yes, benzene with 2 OH.
No, it is incomplete answer. Benzene ring with 2 OH that must be ortho to each other. No, para.
Yes.
You mean the
Both-
position?
Yes. Only when both the hydroxyl OH group in the benzene ring, they are ortho to each other, they are known as catechol ring. Do you understand? So if the 2 OH, they present meta to each other, they are not catechol OH. Is it clear?
Yes.
This concept, I want to tell you, it sounds simple, but very, very important. You will realize why this concept is important throughout the chapter. So catechol means it's not just 2 OH in the benzene ring. 2 OH must be ortho to each other. The 2 OH should not be meta to each other, should not be para to each other. Okay. So this is known as catechol OH. So see, they contain catechol group and also an alkyl amine group. Why alkyl amine? If you look at the side chain, they contain amine group. Okay, side chain. So we go to the next slide. This slide we'll discuss the binding site of the adrenergic drug, that's the adrenaline, to the inside the receptor. Now I want to tell you this slide is the backbone of the whole lecture. Try to understand this slide very well. I will spend a lot of time in this slide. I will expand this. Okay, can you see this now properly?
Yes.
Yes.
Good. Now, I want to ask you one question. How many types of binding interaction you see in this diagram? Try to understand very well. I may finish my whole lecture in this slide itself. So listen carefully. This slide is the backbone for whole chapter. Try to understand. Okay. How many binding? Okay, let me ask 2 questions. First of all, how many binding interaction? And how many? Second question is how many types of interaction you see?
5 interactions and 2 types of interaction.
5 interaction? Is it 2
And
type
2.
or 3 type?
3
3
types.
types.
What are those 3 types?
The one between
Which
nitrogen and oxygen. The one between hydrogen and oxygen. And there is the between the 2 cycles.
Okay, so what is that bonding you see between the hydrogen and oxygen?
Hydrogenic
bond?
bond.
Hydrogen
bond?
and the lone pair of oxygen.
What is that?
H
What is
bond.
the, yeah, what is the name of this bond between the hydrogen and the lone pair of oxygen?
Hydrogenic bonds.
Not hydrogenic, hydrogen bond. Okay.
Okay, hydrogen bond.
Yeah, it's a hydrogen bond. So how many hydrogen bond do you see in the whole structure?
3.
3 hydrogen bond. Can you name them?
How should I name them? So the OH, both OH here on the SER207 and 204, and there is one between the compound and the ASN 293.
Okay, can anyone? Okay, now see, the hydrogen bonds are formed. Okay, how many? Let me ask another, rephrase my question. Try to answer it accordingly. How many OH group is present in the whole molecule adrenaline? And how many type of OH group is present?
3.
Total 3 OH group. And how many type? Are they same property wise?
I'm confused. I can't tell.
How many types? What do you mean by type? You know, the OH, it can be alcoholic OH when it is open, when it is attached to open chain. It can be phenolic OH, aromatic OH when it is attached to aromatic ring, benzene ring, correct? So I have given you the answer already. So how many types of OH you see here?
2 phenolic and 1 alcoholic.
So how many types?
2 phenolic and 1 alcoholic.
2 types.
2 types.
2 types. Okay. And how many number?
3.
3. What are those? 2 phenolic and 1
alcoholic.
alcoholic. Okay. So see, this terminology is again important. So 2 phenolic means they are also, when we say 2 phenolic, they are catechol OH. They are not simple phenolic OH. Are they simple phenolic OH or catechol OH? They are catechol OH, correct? Aromatic catechol OH. And second type is the aliphatic OH, correct? Why it is not moving? So we'll go slow, try to understand. I'll repeat again and again. And every term is important. So what we discussed so far, my question was how many OH you have? First of all, we have seen there are 3 OH group present in the adrenaline and two types. They belong to two different types. One is alcoholic OH and second one is the catechol OH, which are aromatic in nature. And all the three OH are involved in the formation of hydrogen bonding. But they are involved in the hydrogen bonding with the receptor with a different amino acid. For example, okay, so see both the catechol OH, okay, they are involved in the formation of hydrogen bonding with the two amino acid residues serine 207 and serine 204. Is it clear? Whereas the alcoholic OH is involved in the formation of hydrogen bonding with ASN 293, asparagine 293. Is it clear?
Clear.
And
Yes.
yeah, you see here, remember this OH, this hydrogen bonding has different role to play. So when you go to exam and if I ask you question related to hydrogen bonding, you must have to mention which type of hydrogen bonding, which OH is involved. Otherwise, if you say, for example, if you give an answer like, okay, the activity is because of the formation of hydrogen bonding between OH and all. So when you mention OH, you have to mention the type of OH. Is it the catechol OH or is it the alcohol OH? Okay. So with this, we move on next. Now you see, if you look at the benzene ring, okay, and regarding this alcoholic OH, I want to tell you one thing. This must remain above the plane in order to form hydrogen bonding with the ASN 293. What did I say about the alcoholic OH?
Must be above the plane.
Must be above the plane. How do you draw above the plane and below the plane? By a bold line. Bold line means you see this O and carbon bond is bold. It means above the plane where you can see. If it is dotted line, then it is below the plane. Now, same thing I will show you here. I will go back. Do you see this structure? All of you?
Yes.
Yeah, can you see? You see above the plane, okay? You see the OH group means this OH, alcoholic OH, it remains above the plane, okay? As compared to the hydrogen that is below the plane. So for this OH to form a hydrogen bonding with the another amino acid on top, it must remain above the plane. Imagine if the same OH group, it remains below the plane, can it form hydrogen bonding?
No.
No.
But you can feel it, right? By looking at the structure, yes or not? If the OH group, yeah, if the OH group remain below the plane, it will not form hydrogen bonding. That means there are two isomers, remember, R isomer and S isomer. In case of R, the OH remain above the plane, S, the OH remain below the plane. Okay, for the molecule to form the particular hydrogen bonding with the ASN 293, the OH must be in the R form, that is above the plane. Okay, so see how much difference it makes. It will totally go away if it remains below the plane. And you see the catechol OH here. Can you see the catechol OH, which are two OH group? Yeah, two OH group that are ortho to each other, right? I go back to the slide again. Can you see my slide again? Okay.
Yes.
So far we discussed about hydrogen bonding. To sum up, we discussed about hydrogen bonding, okay? And we have seen this hydrogen bonding is shown by two different types of OH group. One is alcoholic OH, and the alcoholic OH must be in the R form above the plane. And another type of OH is the catechol OH group, right? Then you see the third type of interaction is between the benzene ring, okay, with the another aromatic ring, benzene ring of the phenylalanine 290. This is a hydrophobic pi-pi stacking interaction. What is your understanding about the pi-pi stacking interaction? Can anyone tell me?
I have no idea, doctor.
Anyone? Those who have idea? Anyone?
Between aromatic rings.
Yes, it is between. How the pi-pi stacking interaction takes place?
Van der Waal forces.
Not really van der Waal. Okay. What is pi-pi stacking interaction? Tell me. Anyone? Let me draw something here, okay? And I will show you again. Can you see a drawing here? Okay, see here. Can you see the drawing? Okay.
Yes.
So you know the benzene ring, it has 6 pi electrons. Yes or not?
Yes.
So this pi electron, they are stable or they continuously rotate?
Rotate.
They continuously rotate. Because they are continuously rotating, okay, they generates a negative charge at the surrounding, okay, at the periphery of the benzene ring. And they generate a positive charge inside the ring. Do you understand what I said? Because of the,
Yes.
I will draw again. Because these pi electrons are constantly rotating, they generate a negative charge outside the ring and they generate a positive charge inside the ring. This generally I like to explain in the board, but anyway. See. Now can you see this?
Yes.
Yes. So because of the constant rotation of the pi electron, okay, it generates a aromatic ring generates a negative charge at the surrounding of the ring at the periphery and positive charge inside the ring. So similarly, because of this, when two aromatic rings face each other, they cannot face exactly parallel to each other. They have to face perpendicular. That means perpendicular means the positive sign of one benzene ring will face the periphery of the another benzene ring. So they will attract with each other. So this kind of bonding is known as pi-pi stacking interaction. Is it clear?
Can I ask?
Can you repeat it, please?
Yeah. Now, do you understand what is the benzene ring? I explained from the beginning what is pi-pi interaction. You see, because the pi electrons are revolving continuously, you know, due to resonance, the pi electrons are not static. They are continuously revolving. Because of the revolving of the pi electron, it generates a negative charge outside the benzene ring at the periphery, okay? And it generates a positive charge inside the benzene ring. Do you understand? So because of this, when two benzene ring they face to each other, two aromatic ring, they come closer to each other. They will form pi-pi stacking interaction, where the negative charge at the boundary of one benzene ring will be attracted to the positive charge, okay, inside the another benzene ring. This is how they got interacted with each other. Is it clear?
Yes, thank you.
Clear.
Are you sure?
Mm-hmm.
So you see, so we call it as pi-pi stacking interactions. So see, we have discussed here two different types of interaction. You see the periphery of one ring is facing the center of another benzene ring. So in this molecule, how the adrenaline is interacting inside the binding site of the receptor. Okay, so we have seen hydrogen bonding. Now we see pi-pi stacking interaction, which is hydrophobic interaction. And then you see the nitrogen is also forming an ionic interaction with the carboxylic group of aspartic acid. All right. This is the third type of interaction, ionic interaction. And if you notice, the nitrogen atom is attached with a alkyl group. What is the name of the alkyl group here? Hello.
Methyl.
Methyl group, right? So regarding methyl group, you need to remember very important point that I will discuss in the next slide. This alkyl group is responsible. This alkyl group is responsible for selectivity of the drug. And this alkyl group is showing the hydrophobic interaction. It binds inside the hydrophobic pocket of the receptor and shows hydrophobic interactions. So in the whole molecule, you will see two hydrophobic interactions. One because of the benzene ring with another benzene ring of the phenylalanine 290, and another because of the alkyl group that is attached to the nitrogen. This alkyl group will also show hydrophobic interaction by binding inside the hydrophobic pocket of the receptor. Is it clear, all of you? How many hydrophobic interaction I said?
Two.
Yeah.
What are those type?
The pi-pi stacking interaction and the hydrophobic interaction, the ionic interaction.
No. Again, repeat.
The pi-pi stacking
Nitrogen.
interaction.
And the binding
And,
of the methyl group to the active pocket.
Oh, okay, and the methyl group bind. Okay.
Yeah, methyl group bind where?
With nitrogen.
No, it is attached to nitrogen. But regarding bonding, how it interacts with the receptor?
Hydrophobic interaction.
Yeah, so methyl group, the methyl group can be any other group, any other alkyl group. It can be methyl, propyl, ethyl, butyl, tertiary butyl, isopropyl. So we consider it as a alkyl group. The alkyl group that is attached to the nitrogen atom. 2 points you need to remember. The alkyl group that is attached to the nitrogen atom, okay, interacts with the receptor by interacting with the hydrophobic pocket of the receptor. It gets inserted into the hydrophobic pocket of the receptor. Okay, and this is the first point, hydrophobic interaction. Excuse me. Second point, it is this alkyl group which is responsible for selectivity of a drug. Do you understand? Now the question is how the alkyl group impact the selectivity. That we will discuss in the next lecture, not today's lecture. So if you have clear idea, now can I ask you the final question before I move on to the next slide? As I said, this slide is very important. It's the backbone for the whole chapter. If you understand this, you will automatically understand the rest of the slides. Now, how many interaction we have studied so far and how many types of interaction we have studied so far?
We studied 3 types of interactions.
What are those?
The OH interactions, okay, and the benzene ring, and the alkyl group.
No, see, interaction between the two benzene ring and alkyl group they are same, the hydrophobic interaction. Okay.
Two types.
Two different hydrophobic interaction. There is a third type, ionic interaction between the nitrogen and the carboxylic group. Can you see that ionic interaction?
Yes, doctor.
Okay, this is I draw here. This is the ionic interaction. This block, so 3 types of interaction. And again, like in hydrogen bonding interaction, we have seen 2 types of OH groups are present. That is catechol OH in the benzene ring forming the hydrogen bonding interaction with the serine 207 and serine 204, and alcoholic OH which is interacting with the ASN 293, and it must be above the plane in R enantiomer form. Okay. Then 2 types of hydrophobic interaction we have seen. One is pi-pi stacking interaction between the 2 benzene ring, and hydrophobic interaction between the alkyl group, that is the methyl here, and the receptor pocket. Okay, there is a pocket. Assume there is a pocket. So the ring I drew here, imagine this is the pocket of the receptor. And what is the important point I said about the alkyl group? How many of you remember?
It's make the selectivity for.
It is
Responsible for
Important
the
for
selectivity to the receptor.
important for selectivity. So the type of alkyl group decide the selectivity, imparts the selectivity. How we will see in the next lecture. So with this, we will forward to the next slide. So regarding the SAR, okay. Regarding the SAR, I go back. Go back to the slide. This is something a problem when we have to teach, not able to draw. Okay, here. So remember these points. Structure activity relationship, that is phenol group form hydrogen bonding at the binding site with serine 207 and 204. Okay, especially inside the beta adrenal receptor. The phenol can be replaced with other hydrogen binding groups as well. Remember this point, we will discuss in detail about it. Aromatic ring form van der Waals interaction. What is the type of interaction? Pi-pi stacking interaction in the binding site with phenylalanine 290. Alcoholic OH form hydrogen bonding interaction with the binding site with ASN 293. Alkyl ammonium ion forms ionic bond with the carboxylic group of ASP 113, and the N-alkyl group affects the target selectivity, right? And this alkyl group, the larger N-alkyl group lead to selectivity to beta adrenal receptor. Why and how, that we will discuss in the next lecture. Now we move on to the next slide.
Can I ask, Doctor?
Yeah.
Can I ask a question before moving?
Yeah.
What we were binding to the receptor? That compound, is it a drug? Is it another receptor?
It is the structure of adrenaline.
Ah, okay.
It is the structure of, okay, it is the structure of adrenaline, which is binding to the, I'm showing the binding of adrenaline to the beta receptor. Okay. You see here beta adrenergic binding site. So how the adrenaline binds. Then we will see how we developed adrenaline, which is a natural compound that is present inside the body. Okay. So if a patient is suffering from asthma, you need more adrenaline or more adrenaline-like compound, which can activate the receptor, which can bind to the receptor similar way, which can activate the receptor, okay, and help to open the airways. So we are discussing the development of agonist drug. So to be able to understand the development of agonist drug, we first must understand how the natural compound that is present in the body, this adrenaline, how it is binding inside the binding site. So that's why I said this slide is very important. If you do not have clear idea how it is binding inside, we will not understand the development of drug, how the chemical modification will help to develop a better drug. Because when you study the same thing in pharmacology, pharmacology will only tell adrenaline binds to the receptor, beta 2 receptor. It will never tell you how it binds, how the different functional groups are responsible for binding. This is what you study in medicinal chemistry. Okay, so we move on, and you will enjoy this chapter as I move on. So beta agonist in the treatment of asthma. So we have already discussed this, I guess. The beta agonists are the most useful adrenergic agonist in medicine today. They are G-protein coupled receptor. They bind to G-protein coupled receptor. Excuse me. They activate the generation of the cyclic AMP, and their predominant receptor. This beta receptors are predominant in beta 2 receptor predominant in bronchial smooth muscle, and activation of beta 2 receptor result in smooth muscle relaxation, opens the airways, and agonist for beta 2 adrenoreceptors are potential for the treatment of asthma. This can also be used to relax smooth muscle in the uterus to delay premature labor. Okay, but anyway, we'll not concentrate that. We'll be concentrating on the development of anti-asthmatic drug as a beta agonist. Now, this slide onward, our discussion for drug starts. So from nature, the hormone adrenaline, that is epinephrine, was isolated initially, okay, as a glycerol extract from the sheep adrenal. But this drug was found to have poor oral availability, and the drug was administered by inhalation, and it appeared to agonize or trigger the F-beta adrenoreceptor. Okay, so remember, even though the initial days when the drug could not be synthesized, they didn't know how to synthesize in the lab. They were extracting the adrenaline from the sheep adrenal, but this drug could not be given orally because of the poor oral availability. Why it has poor oral availability, we will discuss. We'll see the reason. So this caused bronchodilation, helping people to breathe, okay, during the asthma attack. So there was a huge demand for more drug. To fulfill the demand, they synthesized and tested both synthetic version and the natural product that is extracted from the sheep adrenaline. But the natural drug was found to be 2 times more potent as compared to the synthetic version. Do you understand what I'm trying to tell you?
No.
Then why don't you ask me?
They're fighting outside.
Yeah.
Can you repeat, please?
Yeah. So what I said, what did you not understand? First you tell me.
What is it?
What is that you did not understand? You tell me so that I can explain again.
After extracting the adrenaline from sheep, I lost the voice there. I couldn't hear anything.
Okay. Someone lost voice, someone lost the place, maybe for a coffee or chai, someone, okay. It's all right, but you need to interrupt me. If you do not interrupt me, because I cannot see your face, I don't understand. I repeat again. So see what happened. Initially, this adrenaline was extracted from the sheep. Sheep, you know what is sheep, right?
Yes.
So the natural, what is the natural origin of adrenaline? Initially,
Glycerol extracts from sheep adrenals.
yeah. So they were using from the sheep. But what are the disadvantages? What is the disadvantage found to have with the adrenaline? One of the disadvantage.
It's
Or
for
oral
orally
availability.
available.
Yeah, poor oral availability. Why? I have not yet discussed. That I will discuss later. Okay. So one of the disadvantage, poor oral availability, and how it was given?
Inhalation forms.
By inhalation. Okay. So success of this sheep adrenaline, and there was a huge demand. To fulfill the demand, what they did, they synthesized in the lab. They tried to synthesize the adrenaline in the lab, but they found the synthetic version is less potent compared to the natural, or in other words, the natural. You see, now we are talking about the two sources of adrenaline. One natural, that is naturally taken from the sheep adrenal, another which is synthesized in the lab. But the question here is the natural drug that is obtained from the sheep adrenal was found to be 2 times more potent as compared to the synthetic version. Is it clear? Do you understand the question? Yes or not?
Yes.
So
Yes,
see
the
now,
natural
yeah,
is more potent.
yeah, natural is found to be, was found to be 2 times
Two
more
more times
potent,
potent.
2 times more potent than the synthetic. Now the question is why, right? The question is why. What is the natural here?
Because the nature is R racemic, so the OH is
No. Yeah, so now you see the story start from here. If you understood the previous slide, what it says, the natural is the R form, whereas the synthetic form was found to be a racemic mixture of R and S. So what does it mean, R and S? Because if you remember, the potency depends on the hydrogen bonding of this OH group, the alcoholic OH group. Can you see that alcoholic OH group? Why it is important? Why I discussed so much? It must remain above the plane. Only in the R enantiomer it remains above the plane. Do you understand?
Yes.
sir.
Yeah, but in the synthetic version, it was found to be a mixture of both R and S. In case of S, why the S enantiomer? If I ask you a question, why S enantiomer of adrenaline is less potent? What is the answer?
Because it
Because
won't
the
bind
OH
the
is
same
not above
function.
the plane.
Yeah, answer it correctly.
Because the OH in the alcoholic OH is not above the plane.
Very good. You see, okay. Can anyone try to answer the same?
I said in the S form, it will be distant too far to bind to the ASN 293, as it is in the R form. So binding will not occur.
I will give you 0. Can anyone tell me why I will give you 0?
Thank you.
Why I will give 0 to Osama? No, I'm serious. I am telling you. You will enjoy when I give the answer. I will give the reason. Correct answer, wrong way of expression. All of you know the answer, but you don't know how to express it. Try again. I will give mark to Basil.
Doctor,
Now reflect what Basil said and what you said and how you said. Yes.
it is inactive because it is alcoholic, comes under the plant, and so can form
Under the plane.
hydrogen bond with the acetylene.
Okay, now Osama, can you tell me why I said I will give you 0? What is the mistake you did? Or can anyone highlight what mistake he did? Actually, I like if somebody does mistake. It helps the entire class to explain. Anyone?
We don't know.
Okay, maybe
In
I
the
ask
alcoholic
Osama.
form, the OH was
Yeah.
below.
We didn't hear him.
Okay, so see what Osama said. Can you repeat, Osama, your same answer? Actually, I'm thankful to you for answering like this. I always expect somebody to answer like this. How you exactly answered previously?
Because in the S form, the OH will the distance in the S form of the OH will be far from the ASN, so it can't form any bond, as it is in the R form.
So see, it is wrong. Why it is wrong? How many OH is there?
There's 3.
How many types of OH? So when you say
There's 2.
in the S form, OH will form. Does it make sense?
No.
He didn't.
So who will mention? How do I know when I correct the answer paper?
Which OH?
I have to say alcoholic OH.
Of course. Otherwise, how do I know? You mean alcoholic OH or you mean the catechol OH? That is why I said again and again, you have to remember the type of OH. Every type of interaction of type of OH has different meaning. Different about that. So whenever you answer, if I ask which enantiomer is more active, which enantiomer is less active, R is more active because the alcoholic OH in the R enantiomer remain above the plane and is able to form hydrogen bonding interaction with asparagine 293. So if you do not mention this alcoholic OH, this is the key word, okay, you will get, or you can say the secondary OH. This is also a secondary OH, okay, alcoholic secondary OH. So you will get, you may not get the full mark. Definitely you will not get full mark. You may not get the mark at all. So this is why I'm asking this question. I knew that you know the answer, but you will answer it wrongly. So read this sentence now. The synthetic was a racemic mixture, racemic compound, okay, which had 1:1 ratio of R and S enantiomer, whereas the natural is active R form. This is why natural is 2 times more potent as compared to the synthetic. Is it clear now?
Yeah, doctor.
Okay, now see the next slide. Come here. Look at this. So natural is active R form, while synthetic it contains 50% inactive S form. So if I ask you in the exam, all of you, remember, if I ask you in the exam, which of the following enantiomer is more active or active enantiomer for the respiratory drug? How do you remember? R or S?
R.
How do you remember? Any clue? Like I said,
It's for synthetic and R, it's natural, which there are in fourth.
okay, I know now you remember, but in exam you may get confused between R and S. Which one is active? How do you remember?
Like correct has
S.
R less
See,
S.
we are talking about respiratory, right? Respiratory, R for respiratory. So R for right. Is it clear? There is no science behind it, just a way to remember. Does it make sense? If I say R for respiratory, R for right.
Yes.
Okay.
Yes.
Yeah. R for respiratory drug. Okay. R for right answer. So this is the way you remember. Then you know rest of the logic. So just like 2 lungs, 2 beta 2, 1 heart, 1 beta 1. So just like that, you also can remember this R for respiratory. All right. Now you read only R enantiomer. Only in R enantiomer the alcoholic OH can form hydrogen bond with the receptor, but in S enantiomer it cannot form because the alcoholic OH remain below the plane. So there was a need to find a way to obtain R to meet the demand. So resolution of enantiomer was carried out by converting them to diastereoisomers. So just to remember which one is active and which one is why it is active. So the potency you see, the mainly it is influenced by the alcoholic OH group. Now again, can you solve this question? I have a practice question for you. I already gave the answer. I'm not supposed to give the answer. Anyway, see, write the structure of R and S enantiomer of adrenaline. So see, this is a practice question, but same question I can ask with any drug. How you write the structure? How you make the structure? What is the difference between R and S? How do you draw them?
The O is above the plane.
Yes.
So and the synthetic is down. So I write the OH with the dots.
Very good. Okay. So R means you draw the R with a bold line, and S you draw the OH with a dotted line. Dotted line means away. That means below the plane. Okay. So if you do not write like this, you will lose mark. Be careful. So I can give the structure, any structure instead of adrenaline, any structure, and I can ask. So you need to make changes only in the alcoholic OH. Now explain which enantiomer has greater biological activity by highlighting the interaction of important functional group of the active enantiomer with beta 2 receptor. Can you answer this? Now already we had a lot of brainstorming. Who can answer this correctly?
The R and the R form, the R adrenaline, and the part that's gonna bind is the alcoholic hydroxide group, the OH.
Okay. Answer completely.
Binding it to ASN 293. The binding between OH and ASN 293.
Why? You missed one point again.
What did I miss?
Yeah, think. Yes, because in our enantiomer, the alcoholic OH remain above the plane and can't. So what did you miss?
Okay.
Yeah, why R? Why not? What is the reason behind it? The alcoholic OH remain above the plane.
Yeah.
See, I said always one thing, you know the answer, not only you, most of the student I tell you know the answer. But again, knowing the thing and expressing the same thing, they are two different art. It comes by practice. When you write more expression, of thoughts in writing. But I like you, I consider you as a guinea pig of my class. I do a lot of experiment with you. I hope you don't mind, but I like you very much. I want to tell you, okay? So don't feel offended when I ask you and interrupt you many times. Okay. So you know the answer. Anyone would like to try your best answer last? I think I can stop here instead of going to the next. Should I go to the next slide or stop here?
Stop, please.
Okay.
Okay, so I can stop here, but I want someone to give me the perfect answer. Explanation without missing any keyword. Just make it try. You don't lose anything. Yes.
Natural is more active because the OH, alcoholic OH, is above the plane, and in F synthesis, it's under the plane.
So again, you missed above the plane. So what happened by remaining above the plane?
So it cannot bind with the.
When it remains above the plane or below the plane? You see,
below the plane.
You see, this is the problem, if people always keep half of the word inside, you chew half of the word inside, try to vomit out everything, every point. Now try again. What you missed, you said above the plane, then you stop. So what? Above the plane, it can, because in our enantiomer, the alcoholic OH remain above the plane and can form hydrogen bonding interaction with the important amino acid residue. What is the name of the residue? ASN 293. Now, can you club this keyword? You see the keyword R enantiomer, alcoholic OH, next keyword, then third keyword above the plane, fourth keyword is the hydrogen bonding with which amino acid? Does it form hydrogen bonding with all the amino acid? There are different amino acid. Does it form with all the amino acid?
No.
No, right? With the particular amino. So see, every keyword is important. Now, can you club this all keyword and can give me best answer? Let me see. Last try. Who want to try? Think in your mind and just add these 4 or 5 keywords and answer it.
I want to give it a try again.
Yes.
Okay. The R form is more biologically active than the S form, because in the R form the alcoholic hydroxide is above the plane, and it can form the bond with ASN 293 of the beta receptor.
Yes, it can form hydrogen bonding. Also remember, don't say bond. There are different types.
I wanted to say hydrogen bond, then I stuttered.
No, don't stop. Hydrogen bond is important. What type of, there is a ionic interaction, hydrophobic interaction,
hydrogen bond with the ASN.
Bond, yes, hydrogen bond is also an important keyword. How it interacts, that important. So how do you feel yourself after answering now? Let's compare when you compare your first answer and last answer. Tell me honestly.
A lot better.
So do you agree when I said first time I will give you 0?
Completely.
Okay. So, but I tell you, at the end, you are the one who get benefited, not me. Because when you do mistake repeatedly, you become so firm, solid, perfect. You will never do mistake. So see, one concept, I can ask the same question with different examples of drug. The concept remains same, example varies. Right? So I hope you enjoyed this lecture today until here. Next lecture onward, we will discuss the various examples, how different drugs were developed by overcoming the side effects, improvement, and what are the disadvantages involved with this drug. Everything we'll discuss in the next slide, next lecture, I mean. So we have quiz on next Wednesday, correct?
Yes.
Yeah, we decided on next Wednesday for diuretics, site 1, 2, 3 for 10 marks. Do you want to make it for 15 mark?
Yes, if other students will not.
Majority did not agree to the 15 mark.
Yeah, I know. That's why I'm asking again, re-asking. Think of it. Go for voting. Other class, other section, they agreed for 15 marks. I'm giving them 15 marks. So
Because they already have an exam before. They already had a quiz, so they can't rely on the other one. That's why we're not agreeing to the 15 one. That's my opinion.
oh, okay, I see. Anyway, I will follow to you people as you wish, 10 or 15.
Okay.
Okay,
Well,
then
let's see.
I finish my lecture here. Wish you all have a nice day. Okay, take care and be safe.
too. Thank you.
Thanks. See ya.
Any doubt before I leave? Any question? Any doubt?
Yes,
No.
about the lectures records.
Yeah.
Did you upload them somewhere?
Yeah, it is available. It is available in the team. You just have to download. I have renamed them every part wise, part 1, part 2, part 3. Because if I download, I cannot upload it to your Google Classroom. My server fails. I don't know why. But it is available to all of you. Why don't you download and share with each other?
I think we are not allowed to download them. It's just for the doctors.
No, I made it allowed, accessible to all. You could not download any of the lecture?
Yes, I tried, but I couldn't.
She
How
needs
about
to
the
get a stream app. لازم تنزلي اب اسمه ستريم مايكروسوفت ستريم.
نزلته بس قال لي لازم بس الأونر والأونر هن الدكاترة ما زبط معي.
Can Basil explain them how to download this? You can explain in Arabic. You can type it here.
أنت زبط معك تنزلهم من الستريم؟
عادي نسوي داونلود. هيهم عندي اوفلاين على التلفون.
أنا
ما بعرف ما زبط عندي أنا. هو أحسن شيء لو بيعمل قناة تليجرام بس ما عم يرضى ما بعرف.
جربي مرة ثانية.
أوكي.
كيف يعني انت سويتي حكيت له داونلود واعطاكي انه نو؟
إيه